Let's show you the 3 different types of receptor proteins we see in cell communication. This type of reception is called a G protein couple receptor. And this whole type of signal transduction is going to be mediated by this G protein. Initially what happens is a ligand is going to bind to a receptor protein, which changes its shape. This G protein is going to bind to this receptor protein. Now initially when this G protein binds to the receptor, it's going to have this molecule attached called GDP. It's going to change GDP with a GTP. I know that sounds really confusing, but just follow me here. After this GDP molecule is replaced with GTP, this G protein is going to phosphorylate another protein. Being phosphorylated, this other protein is going to relay that message forward. So basically, these G proteins are going to act like an on-off switch for different pathways within the cell. Our next type of receptor molecule is called an RTK, or receptor tyrosine kinases. In this type of receptor, we actually have 2 receptor proteins that are going to bind together, forming something called a dimer. What happens in this process is the ligands will bind to these receptor proteins, bringing them together. This will phosphorylate portions within the cell and activate certain pathways. This phosphorylation doesn't occur until these proteins come together. Once these tyrosine portions of the receptor proteins are phosphorylated and other proteins bind to them, they're going to send that message throughout the cell. So again, this is just another way we relay a message into the cell. Our last type of receptor protein is called a ligand-gated ion channel. And basically what happens is we have a ligand that's going to bind with a receptor protein. Once the ligand binds to the receptor protein, it's going to open it up, allowing the ions through. This is basically just a gate that opens and closes for ions.